Hello, I'm Stefan Sultana. I'm part of the patient safety organization at AstraZeneca, and I'd like to present to you a couple of case studies where we've used the recently qualified biomarker panel and composite measure in our phase 1 healthy volunteer studies. The first case study is a multiple ascending dose phase 1 study. Compound A is a peptide drug candidate which we're developing with an oral formulation that has to include permeation enhancers in order to optimize systemic exposure. Mild to moderate renal tubular injury were found in 1 and 3 month dog studies, but not studies of shorter duration. And an additional 4-week dog study confirmed that the tubular injury was actually associated with one of the permeation enhancers in the formulation of the drug, not the drug itself. This slide shows the 3-month dog study for this particular program and shows the biomarker pattern for NGAL and clusterin, as well as albumin, which was collected as well. The study was designed to test the different excipients which are part of the formulation for this particular program. And you can see in the bottom left-hand corner the different groups for the study and the different excipients tested, with the red, amber, green markers there showing the presence of moderate to mild to no renal toxicity against each of the legs. If you then compare that to the changes that we're seeing, you can see that for NGAL especially, but also for clusterin, and to a lesser extent for albumin, we're seeing movement in the groups V1, V2, V4, and V5, and all of these groups, in fact, contain the PEB, permeation enhancer B, which is one of the excipients that we think is linked to this particular toxicity. So the biomarker patterns for NGAL and clusterin are correlating quite well with the histology findings. We then went ahead and designed a multiple ascending dose. We'd previously run a single ascending dose without including the biomarkers because they hadn't been qualified as a panel yet. But by the time we came to design the MAD study, the biomarker qualification had taken place, so we could therefore include the biomarkers in this particular study. The study was conducted in 2 separate cohorts, each consisting of 15 subjects with 12 active and 3 placebo per group. Now, both cohorts got the same doses of drug and excipients. It's only for operational reasons that we actually split them out into 2 groups. But from a biomarker point of view, it's actually interesting because it's a story in 2 halves, because we've got very different patterns in the first and the second group that we ran, even though they got the same doses of compound A and the same amount of excipient B in that formulation. We had so volunteers were dosed for 28 days. Placebo subjects received placebo for the duration of those 28 days. The 12 active subjects were dosed for 7 days with the first dose level of compound A, and then after 7 days they escalated up to double that dose. After a further 7 days they escalated up to double that again, so we're up to 4 times the dose of compound A. And for the last week of the study, subjects escalated with a doubling again to 8 times the dose of compound A, receiving the same amount of excipient B throughout the study. So the content of excipient B was kept the same regardless of the dose of compound A. And we collected, it's a 28-day study, right? So it's quite a long phase 1 study, which is actually unusual in that regard. And we collected samples for urine samples for biomarkers pre-dose at 24 hours post-dose, and then approximately twice a week until 29 days, i.e. the day after the last day of dosing. Now, this slide shows us the composite measure results. Your left-hand side shows you in tabular form, and the right-hand side shows as a box and whiskers plot representing the median and interquartile range of the individual composite measure results for this particular group cohort. For the threshold for this composite measure, we chose a 5% threshold, and because we have an imbalanced design, we have 12 subjects on active and 3 subjects on placebo, we chose the no placebo comparison group for the thresholds, which gives us a composite measure threshold of 1.21 to aim for. So anything above 1.21 would be a red flag. Now you can see in group 1 that we found out quite quickly that quite a few of the measures that the results that we were getting back were above the threshold for this particular composite measure. And we looked at the individual biomarkers, and we saw that it's not just one biomarker that's pulling up the results. It's actually quite a few of the biomarkers were showing quite a significant fold change from baseline in pattern across the 28 days. Now, to tell you a little bit about what's happened with this particular dose cohort, this is the first time we're using this biomarker panel in a phase 1 clinical study, and the team decided to test the pre-dose samples and the day 18 samples together as a single analysis batch in order to inform whether to start that second dose cohort that we're preparing for, which was receiving the same doses. We wanted to inform the start of the second dose group using some data, but not all the data from the preceding group. And we had time to test the pre-dose and day 18 samples. And those came back quite normal. If I go back, I can show you that day 1 and day 18 showed absolutely no difference in value. And even you look at biomarkers, they're very well behaved across the whole panel. Day 1 and day 18 seem to be outliers in that they're very well behaved biomarker patterns as well. We then tested the other samples collected, day 2 to day 15, were tested in 2 further separate batches, and the day 21 to 29 samples were tested in a further 2 analytical batches. So one of the problems that we thought of here is that we might be getting batch to batch variability in the results that we're getting, depending on which analytical batch the samples came from, because we only tested the pre-dose samples once with the day 18 samples. All the other timed samples were tested without a baseline sample in that particular analytical run. We looked at creatinine and urinalysis for any proteinuria hematuria signals, and we saw nothing in the routine safety measurements. So we thought, this could be an artifact in the data. Shall we go ahead and carry on and go ahead with group 2 and analyze the data in group 2 and see what we have? And that's exactly what we did. And this is why it's a story in 2 halves, because these are the data from group 2. We'd learned from our previous experience, and the group 2 samples were all tested at the same time within the same analytical run, basically. And lo and behold, you can see that same number of subjects, 12 plus 3, 15 subjects in total. Same doses given to the first group, but this time all the biomarkers, composite measures, all the time points are below the threshold. In fact, they're not even exceeding 1. And looking at the spread across the whole group, it's again, there's no change that we can see across time for this particular composite measure. And the same goes for the biomarkers themselves. They're all very much more behaved. They're all flat and showing no significant trends over time. Each of the 6 biomarkers is behaving quite well indeed. So our overall conclusion is that looking at group 2, we have a uniform pattern across the 28 dose cycle with no difference between placebo and active groups. We're still not clear why the group 1 samples seem to have those aberrant results, and we think that there's some batch effect going on with the biomarker levels. And for us, at least, this particular study showed the importance of testing all samples from individual subjects within the same analytical run as far as possible. We have a second case example, which we'd like to share with you as well. Our second case study is Compound X, which is an antisense oligonucleotide drug candidate. And again, we're going with an oral formulation for this particular program, and that includes permeation enhancer Y. We saw mild to moderate renal tubular injury, degeneration and necrosis with partial reversibility in 8 and in 12-week dog studies. Shorter dog studies up to 6 weeks duration did not show any pathological changes. We did a separate preclinical study to assess whether the injury is linked to the drug, the ASO, or the permeation enhancer, and this study confirmed that what we're seeing is likely linked to the enhancer Y. This slide shows the biomarker pattern of the canine study. Similar to the previous example, your bottom left-hand panel shows you the different groups studied in this experiment, with group 1 being vehicle only, group 2 being vehicle plus the high dose of the formulation excipients, and then groups 3 to 5 are ascending doses of both the compound and the excipients that go with it. And again, your green, amber, red color code there signifies no toxicity versus mild versus moderate renal tubular injury toxicity. If you look at especially groups 2 and groups 5, which are the two groups that received the high dose of the excipients, these two groups showed a good biomarker response when you look at clusterin and NGAL, but not so much when you look at KIM1. In this particular study, we also looked at albumin and looked at beta-2 microglobulin within the same study toxicology study design. And these two biomarkers also showed a response to the injury, perhaps reflecting that outside of the biomarker panel of 6 biomarkers, there are other injury markers that can also be useful from time to time. We took this biomarker panel and tested it in our single ascending dose study, first time in man. It was carried out in male normal healthy volunteers. There were 3 cohorts planned and conducted for this study. Each cohort consisted of 6 active and 2 placebo subjects, so there are 3 dose cohorts. It's important to note that our placebo group did not receive any of the drug, any of the ASO, but actually the placebo itself contained a mix of 1 to 3 times the amount of excipient Y in that particular leg of the study. And so, for example, the 2 placebo subjects which were in group A, which accompanied the 6 subjects receiving dose A, they got 1 times the amount of excipient like the dose A subjects did. The 2 subjects in dose B got twice the amount of excipient only, similar to the dose B subjects. And for dose C, these 2 healthy volunteers got 3 times the excipient in their placebo arm, similar to the active who got 3 times the compound and 3 times the excipient as well. So it's important to note that in this particular study, we don't have a true placebo arm. We don't have subjects who received no drug and no excipient either. We collected urine for biomarker analysis, And this time we collected it pre-dose, and it's a single ascending dose study. Subjects were sent home at the end of the third day post-dose. So they got 12, 24, 48, and 72-hour post-dose sample collections before they were discharged from the phase 1 unit. Here's a table showing you the phase 1 composite measure results grouped by dose group and by time point of sample collection. You got placebo, dose A, B, and C in order, and you got the time points on your left-hand side. Now we can see from this table, we also have your third column is the composite measure of the group itself, and the fourth column is the composite measure of the active arm divided by the placebo arm. Now, if we want to look at excipient Y as being the target substance that we're interested in, we would need to look for the thresholds in the third column and ignore the fact that we have a placebo arm, because obviously the excipients are also present in our placebo group. So therefore, we don't have a comparator group for that particular evaluation. So we look at these particular figures in the third column for our analysis. Whereas if we were worried about ASOs, now remember ASOs are one of those drug modalities which can sometimes cause kidney injury, and they're not always predicted by preclinical toxicology data. If we're interested in looking at ASOs as our target compound that we're interested in, then we look at the fourth column where you're dividing your composite measure for the active dose by the corresponding time point for the placebo arm. So you're looking at a composite measure ratio of active divided by placebo and using the relevant table in the qualification document. And down here, we've got the 5% threshold level, and I'm showing you that the no placebo comparison would use a threshold of 1.27, whereas the comparison to placebo for that particular group would be a threshold of 1.31. And I've highlighted in red the numbers which exceed this threshold in this particular evaluation. Similar to the previous case example, we plotted the composite measure for the individuals as a box and whiskers plot, and you got the median and the interquartile ranges displayed on this particular graph. And the red bars are your placebo group, which have the mixture of the excipients, remember. Your green boxes are your darker green boxes are the first dose level, lighter green is the second dose level, and purple is the third dose level for drug and for excipients. And similar to the table that we've seen before, you can see that for placebo, but also for the higher dose levels, especially at 48 and 72 hours, we are seeing some movement in our biomarker levels. So this might signify that there's something going on at the injury level. And these are the individual biomarkers showing it's not just one biomarker doing this. There's actually 2 or 3 of them moving in that same direction. You've got clusterin, cystatin C, you got KIM1 showing some movement, and even NAG is showing a trend as well. So it's not just one biomarker, and therefore potentially pharmacology acting here. It's across different biomarkers. Based on the results of this particular study, we decided that we're going to have to repeat further studies looking at permeation enhancer more closely and look for different formulations in order to be able to deliver an oral delivery of our particular ASO. So this study actually helped us to make a decision based on the PK systemic exposure of our drug combined with the renal safety profile that we saw especially for the top dose that we were testing, made us go back into the labs to look for a new formulation and therefore helped in decision making with this particular program. Thank you very much.
Hi, my name is Glaab. I'm at Merck & Company in the Safety Assessment Group in West Point, Pennsylvania, and I'll be talking about the translational kidney safety biomarkers that we use in drug development and a specific case example. So we've actually used these kidney safety biomarkers over the last 10+ years to really improve drug development and the ability to monitor any potential liabilities through the development, both non-clinically and into the clinical setting. We've used these biomarkers for a variety of different reasons, right? And we use it not only to demonstrate that we can monitor the drug-induced injury, also demonstrate reversibility of such injury, but we also use it a lot for guiding different investigational programs, de-risking backups. So it's more than just taking that one molecule and moving it forward. We do use these kidney biomarkers for a variety of different reasons. One particular example, though, really does stand out as a beneficiary of these particular safety biomarkers to monitor kidney injury, right? So we've had multiple programs that have had toxicity in different segments of the nephron, not only in the proximal tubules, but we've had some programs with glomerular injury as well as distal tubule. And we've used a panel of different biomarkers to track these particular lesions, and not only in that particular compound, the lead compound, but also for bringing some of the backups forward. So I mentioned Relebactam. So this is the example that I'd like to go through today. This is a beta-lactamase inhibitor. It was being developed as a fixed-dose combination therapy with Primaxine, and there's 2 additional antibiotics. And the indications for this particular combination therapy was for some pretty severe infections, right? So hospital-acquired and ventilator-associated pneumonia, as well as some of these complicated intra-abdominal infections and complicated urinary tract infections, right? So it was always being developed for this combination therapy with these 2 other drugs, and really for these kind of hard to fight infections. But what we noted when we were developing Relabactam was that in some of the early non-clinical studies, we had kidney toxicity findings, right? The rat was clean, right? We didn't have any liabilities noted in the rat studies. However, in the non-human primate studies, we did see drug-induced kidney injury. And we noticed this at a 1-month duration following treatment. And this was pretty severe degeneration noted. And the first and foremost the thing that we did was to actually use urine from those animals to demonstrate that KIM-1 did actually monitor this particular lesion. There was also a recovery group on this study, and we also showed that the KIM-1 returned to baseline, right, below limits of quantitation following the recovery period. So we also knew that the KIM-1 would track reversibility in these particular animals. So what we did is we actually demonstrated this and used these data to really support the translation of this compound to early clinical trials, right? KIM-1 was selected as the only biomarker to use here because it had performed so well in the non-human primates. And I should also note that this work actually predates a lot of the composite measure qualification that you've heard about previously. So only KIM-1 and only really translating into the phase 1 clinical trials. And what we observed when we actually ran the phase 1 trials, it was that no increases in KIM-1 were observed, right? So we ran both a multiple ascending dose study as well as daily dosing studies in different numbers of patients, normal healthy volunteers. And what we actually found was, or what we didn't find, were increases in KIM-1, right? So we were pretty confident at this point that there was no induced kidney liability in humans, and truly leveraged the KIM-1 data to support the advancement of this particular compound into phase 2 and phase 3 trials. Since then, right, so here's just an example of some of the safety and efficacy studies that were conducted in the phase 2 and phase 3 trials. And we won't go through all of these specific examples, but the bottom line here is that a significant number of patients were then treated with this combination therapy. First and foremost, we showed that there were no increased kidney safety liabilities with this combination therapy. And then we also demonstrated actually a superior renal safety profile compared to some of the current therapies out there. So KIM-1 was not measured in these trials. It was just BUN and creatinine. I should note that, right? But it was the phase 1 data that actually allowed us to move into phase 2 and phase 3 with confidence that there would be a lack of any kind of kidney liability in these larger patient cohorts. So just to summarize, right? So the impact of these translational safety kidney biomarkers really helped this compound, Relabactam, not only get through what we noted preclinically, but really leveraging this data to really take this into some of the phase 1 clinical trials. Using KIM-1 in that phase 1 trial, we demonstrated that there was a lack of kidney injury in those healthy populations. And really, it was those data, both the non-clinical and then the phase 1 data, that actually allowed us to really advance with confidence into the phase 2 and phase 3 trials. So the end of the story is the fact that the FDA did approve Relabactam in this combination therapy in July 2019 to treat these complicated urinary tract infections, as well as some of these intra-abdominal infections in adults. So it not only was it successful in translating to early clinical phase trials, but ultimately it helped get the drug on the market and a successful FDA approval. Thank you.